Hi, everyone. My name is Nadia Gray. I'm an environmental and sustainability studies student. And for this semester, I have been working with Dr. Kathleen Hunt of the biology department here at George Mason. And my project is assessing reproductive hormones in adult female blue whales by analyzing historic baleen samples from the 1940s. So let's talk about the blue whale. So this is a marine mammal, and it is the largest animal ever to have lived, as far as we know. It is about as big as 3 school buses lined up bumper to bumper, and as heavy as 25 African elephants in one room. And on average, they can live anywhere between 80 to 90 years. And the reason why we're talking about the blue whale is because despite the fact that commercial whaling has been banned since 1986, this animal is still considered endangered by the IUCN Red List. This is due to other stressors such as climate change. And although their population is increasing, we still have yet to see stable numbers. And that being said, we also don't know that much about blue whale reproduction, and that is because these animals are too large to collect blood samples from, which is how hormone data is generally generated. There have been some studies trying to use other methods such as fecal samples, respiratory vapor, and blubber collections. However, just due to the nature of these animals, it is a very difficult method to collect. We're going to be looking into using baleen as a method. And baleen is the keratin-based apparatus you see growing inside of a whale's mouth, and it is used to filter feed out krill. And for this study, we're also going to be looking at the steroid hormone progesterone. And that is because in high amounts, we believe that progesterone can indicate pregnancy. So for questions, we're going to be looking at, is progesterone detectable in historic baleen? How long is the gestation period of a female blue whale? And what was the number of calving intervals during the mid to late 1940s? The sample size that we looked at in this project consists of 4 adult females, 2 of which were pregnant upon capture, and 3 of which had an intact root. And that is an important piece of data because baleen grows similarly to how teeth grow, where it has a root that anchors into the gums but isn't necessarily visible. So if you were to cut at the gum line, you'd be losing out that little last piece of data. And then this is where we move on to the lab section. So the first step is to collect your samples. So this was done by drilling along the length of each baleen plate and collecting powder samples every centimeter. Next step is to weigh out the powder of each centimeter of the baleen plate. We measured out around 20 grams for each specimen. And then you can move on to the extraction process, which is where you are adding methanol to the powder, and that is what pulls out the hormones. And then the next step is you are going to remove that methanol that has been sitting in the powder and separate it from the powder pellet that you see there. We're going to move on to the reconstitution phase, which this is after you have dried down that previous methanol sample and you are essentially rehydrating it so that you can create samples with it. And then you're going to have to go ahead and dilute those samples because the progesterone sometimes is too high in specimens to be able to accurately read it. And for my specimens, all of them were 1 to 4 dilutions, except for one that was 1 to 40. And then finally, you can move on to the assay portion. So each sample that you have created is going to go into each of these wells. And once you're done with that, it'll look something like this. And then you can place the assay tray into what is called a spectrophotometer. And this brings us to our results. So this is what the raw data looks like. Everywhere you see light colors, that is what is considered to be high progesterone. And everywhere you see is dark, that is what we consider to be low progesterone. And the unit that is used in the spectrophotometer is called ocular density. Data ends up looking like. These graphs are made through a program called Prism. And if you are interested in a more in-depth description of what is going on with each of these graphs, you are going to have to look at my poster of the same name. But essentially, anywhere you see a yellow bar is what we consider to be high progesterone. And on average, we found that these whales were experiencing on average of a 2-year calving interval, with the exception of figure D, which that one only had 2 high progesterone points. And prior to that was all low progesterone, which we later found out was the smallest whale of the population. So this could potentially indicate that she had not yet been reproducing because she might have been younger than the other specimens in this sample. And as for the next steps, the purpose of this research is to generate baseline data for future hormone studies in blue whales. So essentially, we are creating a control group, and by testing whales from other time periods, we can use this and determine what is considered normal reproduction. And then this knowledge can also be used to apply towards future management plans of the species. And then this brings us to the end of the presentation. I would like to thank Dr. Michael McGowan and John Osowski from the Smithsonian National Museum of Natural History. I'd like to thank Ali Case and Dr. Kathleen Hunt over at SciTech at George Mason University, as well as Dr. Allison Fleming and Malia Smith of University of North Carolina, Wilmington, as well as all of the other hardworking scientists and contributors who helped us collect, prepare, and save the specimens. And then finally, I would like to thank the George Mason Oscar Program for allowing us funding to make this project possible. And thank you for listening to my presentation.